In this video, we will discuss about the CAR-T cell anti-tumor efficiencies in the absence of FAS signaling. In the previous video, we have understood if the FAS has been knocked down in the T cells, how it will increase the CAR-T cells anti-tumor efficiencies. And here they validate their same hypothesis by knocking out the FAS ligand gene in the tumor cells. In the previous video, we have discussed they have knocked down FAS ligand gene in the T cells. So before going into the final results, what they have presently shown is that in the absence of CAR T cells, if FSL ligand is been introduced in vitro, these are the in vitro studies. So if FSL ligand is introduced into the cell culture medium, what they have seen that it does not affect too much into this null M6 or Raji cancer cells. However, it will affect the T cells, that we can see that with increase in the concentration of FSL ligand, they will see a decrease in the survival populations of the T cells. So thereafter, they also wanted to check the effect of this FSL ligand if it is knocked down using like different approach into the mouse. So first they inject the mouse with these cancer cells null M6, and thereafter they use the 4 different conditions. First they transduce the normal T cells, then this CAR T cells without intercellular domains, then CAR T cells with intercellular extracellular domains, and then knock down FASL gene using AAV, and finally these intercellular CAR T cells with intercellular and extracellular domains, and knock out this FASL ligand using CRISPR-Cas9 system. And what they are able to see compared to the CAR T without intercellular or extracellular domains, they are able to see that if FSL gene has been knocked down through any approach, they are able to see an increase in the survival rate in both the cases compared to the control. Thereafter, they validate some of the previously published literature where they have shown that this FSL amount goes up with increase in this TNF RFS10B, which is related to the death pathway or trail pathways, this receptor is related to the death or trail pathways, and this Cas8 and FADD is also related to these trail pathways. So these are the results that are published, and they have validated these results using their CAR T cells populations versus control populations. And what they are able to see in the case of control, they do not see too much difference, but when they knock down FAS, they do not see any significant difference yet. But when they increase this TNF RS10B, they are able to see an increase in the CAR T cells, and similarly for FADD and Cas8 also they are able to see increase in the CAR T cells. And this similar results they have seen for in the other publications also. So both together results are also in line with the observations that the authors have seen in this paper, authors that are presented in this paper. And as why the researcher says that, because this TNF RSF10B and FADD Cas8 are usually found high in the tumor cells such as null M6. And thereafter, next, what they wanted to check whether if FSL signaling pathways has been disrupted in the tumor cells, this also has a similar effect in terms of the CAR T cells increased anti-tumor efficiencies. So when they check this thing in the null M6 cells, they are able to see that with the decrease in the effector to target ratio, they are able to see a decrease in the survival populations of the null M6 compared to the case when both of them are cultured independently, that is FASL knockout null M6 and FASL wild type present null M6 are cultured independently, the growth rate increased. However, when they are cultured co-cultured with CAR T with intercellular and extracellular domains, and one without an intercellular extracellular domains. In both the cases, they are able to see a decrease in the LM6 populations. However, the difference between FASL wild type and FASL knockout is not significant at this effective target ratios. But their results can clearly be seen when CAR T cells is co-cultured with the LM6. So this in all together indicates that whether this in the absence of FAS signalings, it does not cause any loss in the CAR T cell anti-tumor efficiencies, as can be seen in these results and also the results that we have discussed previously in some of our previous videos. So if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.